Hello everyone, in this session we are going to talk about the conversion of third butyl alcohol to third butyl chloride through SN1 reaction. So the reaction that we have is including substrate, nucleophile and a leaving group. For that we need a weak nucleophile group which are as you see in this slide. And you know that the nucleophile is the chemical group that it likes the nucleus. So it just participates to be adsorbed in the nucleus. And you see that the rate of the reactions in SN1 is just related to the concentration of the substrate. But in SN2 is related the concentration of the substrate and nucleophile. So that's the important difference between SN1 and SN2. So here you see that there are a table showing differences between SN1 and SN2. For SN1 you need weak nucleophile, but for SN2 you need a strong nucleophile. And as you see, the first SN1 is happening for tertiary substrate, but SN2 happening for primary substrate. And you need a good leaving group for SN1. And also during SN1, we have a carbocation, but no carbocation in SN2. And in SN1, the reaction happened in 3 steps, which is very important. So in our work, we have a third butyl alcohol, which is changed to third butyl chloride. You see how the reaction is. For the mechanism, you see, first of all, we produce oxonium ion, and then after producing oxonium ion, we have production of the water through the carbocation, which is the slowest step of in these 3-step reactions, and this step is the rate determining. After that, we have the attack of the Cl as a nucleophile to the carbon, and then production of the third butyl chloride in the fast step. So here you can see again how the reaction happens, the mechanism again. You need to remember all the steps. First of all, what anion, or what cation you need to have to make the reaction. So you have the first oxonium reaction, then you need to have the separation of the water, and after that the attack from chloride to the carbon plus. Again, you see the mechanism and some explanations, which is good to read. So in this step, we have the chemicals that we are going to use for our experiment. We have the hydrochloric acid, third butyl alcohol, and we have the 5% sodium hydrocarbonic, and we have the anhydrous calcium chloride. So as you see, we need pasture peptides for mixing and just transferring materials. We have it in 2 forms of plastic and in glass form. For the glass form, we need the bulb blubber for just mix for to suck the materials from the test tube out. As you see, there are safety cautious. You need to remember them. So we have the test tube. We add 5 milliliters of concentrated hydrochloric acid. Then we add 3.9 gram third butyl alcohol, and we use the pastures that we seen before to mix these two unreacted materials. As you see, after mixing by the use of pastures, we can make the reaction happen, and the third butyl chloride just obtained on top of the liquid phase. So we need to remove the liquid phase from the test tube. We do that with the pastures that we have, and after that, to remove the unreacted ions, we use water to wash the products. You see that this step. So we add the water, and as you did the same, we use the pasture for just the mixing, and then we let it to be stable, and after that we remove the water from the bottom of the test tube with the use of the pastures. So we add again, we add 5% sodium hydrocarbonic, and you see that the carbon dioxide just goes out from the test tube, and you need to remove the remaining 5% sodium hydrocarbonic. Then we add water for washing again, and repeat the washing step again. So we, at the end of the reaction and experiment, we need to add calcium chloride for drying process. So you see that calcium chloride is not participating in the reaction, just we need it for drying process. So after drying, we just transfer whole third butyl chloride to the round bottom flask for the next step. You see, we need this simple short pass distillation just to distill the obtained third butyl chloride, and we just gather the distilled materials in the elbow. You see that? So these are the different parts of the distillation. We need the Kleisen adapter, and we need to have a thermometer. So perhaps we need to use ice. So we put the ice on through the Kleisen adapter. So after collecting the distilled, we need to determine the yield of the experiment. You see that in the next step. So at the end of the experiment, we need to measure the yield of the experiment. So we need to, the hypothetical yield is 2 gram. We have the molecular weight for all materials that participating in this experiment. We know that the yield is obtaining based on the mole of the product over the reactants. So you need to measure first the mole of the butyl chloride that we obtain, which is 0.0216 over 0.05. That so we obtained 43% as a yield of this experiment.